Broad's a special place because my very first talk in Boston took place at the Broad when I visited Dr. Ann Carpenter and Dr. Shantanu Singh a few years ago. So excited to be back and sharing some of the work that I'm excited about. So I'll get into it. I thought that the part of my research I'd share with you today is what I consider to be one of the most important questions for the field. It's how can we make medical AI, which we know works, and which we know there has been a lot of advancements for, a part of routine clinical care, which we also know really hasn't taken place so far. And I want to break this down into two parts. One is talk about the foundation that we're building on to even be able to ask this question. And as part of that, I'll talk about how we have been able to develop AI that performs as well as experts, and then how we've also been able to study the effect on clinicians with AI support. The second part of my talk will be centered around three other aspects, which is asking how can we make progress towards making AI a routine part of clinical care. And as part of this, I want to argue that there are three pieces we need to think about. One is how do we design effective clinician AI collaboration setups in workflows? How do we build data sets that are reflective of real world tasks? And how do we develop algorithms in ways that allow us to tackle not just a few set of diseases, but really allow us to tackle the broad range of diseases that medical experts tackle on a daily basis? Great, so we'll dive in. I want to start with talking about developing AI that performs as well as experts. Over the last few years, there have been several demonstrations that deep learning can perform comparably to medical experts, and certainly within the domain of medical imaging across ophthalmology, dermatology, pathology. These were some of the foundational early studies in 2016, 2017. And this is some work that was actually my first introduction into the intersection of deep learning and medicine, where we showed that using deep learning applied to single lead ECGs that are recording heart rhythms, we could determine whether patients have abnormal heart rhythms, and then demonstrated that with a combination of good deep learning algorithm development and getting data labeled well and at scale, we could perform at the level of cardiologists on this data. And this was one of my first projects. In 2019, this algorithm actually got FDA cleared and is in use as part of iRhythm's system for evaluating patients with suspected arrhythmias. Another project, which I'll talk more about later, is looking at chest x-rays and how can we do this very common interpretation task. Chest x-rays, there are over 2 billion of these that happen per year, and how deep learning algorithms really over the last 3 or 4 years have demonstrated that they're able to detect multiple pathologies from chest radiographs at the level of radiologists. So this is one of the foundational blocks, which is that we can perform as well as experts. And it's really been driven by advancements in algorithms like convolutional neural network architecture improvements, training improvements, and by large data sets. And this have happened really by, I would say, a focus on improving the pipelines for data collection and for labeling over the last few years. I think when we think about algorithms working in clinical settings, we really don't care as much about whether the model performs as well as clinicians on these sets. We really care about a different question, which is this model is going to work as a decision support for people in their workflows. And let's see how they do versus if those clinicians were unassisted in what I'm going to call a mimic workflow, so not actually taking care of real patients, but trying to simulate what these clinicians are doing in their actual workflows. And here's a very simple setup. So this is some work that we did in collaboration with a few embryologists at UCSF, where we're saying we're going to try to evaluate whether an embryo is going to survive at the end of 25 hours after thaw. And we're going to try to do that 2 to 3 hours. And this is a task that's performed by embryologists fairly often. And the combination experiment that we're going to do is the simplest one we can think of. We're going to ask a deep learning model to say, how long will this embryo survive? We're going to ask an embryologist to do the same, and then we're going to average their prediction. Note that in this setup, the embryologist isn't actually using directly the model output, but both of them are being combined in some way. And this very simple combination setup, you can see that compared to either the model ensemble or the embryologist, if you combine by simply averaging their predictions, you do pretty well. Okay, so this is hopeful. I want to talk about a slightly more interesting assistance setup. So let's look at the tasks of knee MRIs. Here, there are several slices that a radiologist goes through to evaluate certain abnormalities, including whether there's an ACL tear, an MCL tear, or general abnormalities. We asked, what if we showed 3 probability numbers corresponding to these 3 tasks? How would the performance of radiologists change? Oh, by the way, this is a task that's done by subspecialty trained radiologists with an extra 2 years of training required. Can we have general radiologists and surgeons who also do this task, but also send this to radiologists for interpretation, do it as well as subspecialty radiologists can? And so we design an experiment where we say, let's have the radiologist take a look at this, at these cases with model assistance and without model assistance. Let's have a washout period. Let's say how they do. And here's what we found. If I'll focus our attention onto this finding over here, which was our significant finding, is that the specificity on detecting ACL tears went up by about 0.06. And what this corresponds to is for 100 people who don't have an ACL tear, we're now able to tell more correctly that we're able to tell for 6 people more correctly that they don't have an ACL tear. Implication is less surgery required, save costs, et cetera, et cetera. So this is hopeful as well. But you can imagine probabilities showing those to radiologists doesn't allow for real sort of explainability for the clinicians. So let's think of a different setup. And here we're looking at a different modality. Here we're looking at head CTAs. And on head CTAs, we want to be able to detect aneurysms because if we are able to detect aneurysms that are big enough, we'll be able to detect them before they rupture and intervene appropriately. For the AI augmented read in this case, what we did was we integrated into the radiology viewer system by saying we're going to run an algorithm and have it output directly into the software that radiologists use to navigate these scans and highlight the portion that they should bring their attention to. And with this setup, we had a similar setup of saying we're going to have unaugmented reads followed by AI augmented reads and vice versa. And here we showed a more clear improvement, not just in specificity, but also in sensitivity and the accuracy of this particular, for this particular task. So this is hopeful as well, because we're able to show, at least on a retrospective set, that we're able to improve here 3 radiologists, 1 neurosurgeon, and 1 resident on this particular task. So that's hopeful again, but where do we go from here? One thing that's interesting to ask here is who's benefiting from AI and who's not. So this is a breakdown looking at individual radiologists and how their performance changed in terms of sensitivity, specificity, and accuracy. And here in yellow, we have their unaugmented performance, and in blue, we have the augmented performance. So we expect going left to right, yellow to blue. And what we found, and I'll direct your attention to radiologist 4 over here, is there's this evidence for the people who are benefiting most are the people who are not performing that well in the first place. Okay, that makes sense. Here's another dot here, radiologist 5. The radiologist 5 dot pair says the radiologist who was best at the task to begin with, not really having much of an improvement. So in this way, the evidence here is pointing towards that the people who are going to benefit most from AI are those who are going to be the least trained. So now we can investigate that question. Let's take a different modality, just because it's interesting to explore how you have to change workflows based on the task and based on the domain we're looking at. So here we're looking at assisting pathologists. Now, pathology is an interesting world because unlike radiology, where a lot of the digital transformation has happened, that digital transformation is yet to happen in pathology. If you just look nationally within the US, only a fraction of pathology workflows have moved over to digital. And then the question becomes, how do we embed our AI software to be able to solve this problem? So one idea here was if there was some way to get at a small patch that a pathologist would be able to pick up, either by taking a screenshot of a digital slide or by taking a photo of what they're seeing under a microscope, they can upload that onto a website, which will then give them some feedback and some assistance that they can incorporate in making their prediction. Note this is a fundamentally different setup here because what's being accessed by the algorithm is determined by what's chosen by the pathologist. The algorithm doesn't have access to the full information, so the pathologist has to be good enough in also selecting a patch that's going to be representative of determining what cancer subtype this is. Here we're looking at liver cancer. So here's what I call the variable expertise effect. Here's the setup we had first. So in this case, we had a little bit of a set of training cases. So there were 2 training cases in which we're trying to make sure that the pathologists understand the task. And then we're alternating between blocks of assisted and unassisted cases that these pathologists are looking at. Great. And now we're breaking down the performance by group, by specialists in gastrointestinal pathology, non-specialist trainees, and pathologists not otherwise categorized. So trainees is the ones we expect to be the least good at this, and that's reflected in the unassisted number. It's 0.85, and you can see this is where we have the greatest gain. The pathologists not otherwise categorized, we can think of as non-specialists as well. And here's a group in which we found a decreased performance. So that sort of validates this hypothesis that it matters who's using the AI tool. So those are the fundamental building blocks for us to even ask this question of how can we make medical AI? We have not only evidence that we can have medical AI that performs as well as experts, we also have some evidence that in simulated clinical workflows, we should be able to improve clinicians as they're performing routine tasks. Cool. So where do we go from here? I'm going to start by talking about design of effective clinician AI setups. And as an example, I'm going to share one of the studies we wrapped up in 2020, which is trying to get towards what has been, in my group thus far, the most realistic evaluation of AI systems and clinical workflows. And here the setup was, we wanted to have people determine whether a patient has active tuberculosis based on a combination of their lab values and an associated chest X-ray. And by the way, we're looking at HIV positive patients. And by the way, these physicians who are going to be taking a look at these cases are going to be physicians in South Africa who do this on a daily basis on a cohort that's representative of the patients they see on a day-to-day basis. Cool. So here we perform a within subjects intermodal study. kind of reflective of how one might even test an AI tool while prospectively trying to deploy it, where we have some cases that are shown with AI assistance, some cases without, and it's randomized whether or not AI assistance is going to be given for a case. When AI assistance is given, we're going to have an algorithm TB prediction scale from very unlikely to very likely. And we're also going to have the algorithm highlight which region of the X-ray is most consistent with a tuberculosis diagnosis. Great. In the unassisted setup, the clinicians don't get to see any of the algorithm assistance. All they see is the image of the X-ray. You can modify the brightness and the contrast, and these lab values with associated reference ranges. Great. So what we find here, I will also mention, one of our learnings was that if it's an AI tool, we need to make sure that there is training associated with having the physicians use this particular tool. So this time we decided to really go after training as an important component and said, for the purposes of training, we're not going to look at one or two cases. We're actually going to look at hundreds of cases, and we're going to ask the physicians to go through hundreds of cases where as soon as they've entered their prediction, they actually get a feedback on whether their prediction was correct or not. And this is through a microbiological test. What was amazing about this study was we were doing it with 15 or so physicians in South Africa, and they completed this training phase and they said, Pranav, since training 10 or 15 years ago, this is the first time I'm actually seeing whether my predictions were correct. Because that feedback loop is not something that they get very often. They have to decide on the spot, am I going to treat this patient for active TB and not wait for the microbiological test to come back to help guide that decision. So that was fascinating for me to learn and useful in this particular setup, because what I was trying to get at is having them calibrate to whether they should be trusting the algorithm or not, and have them make their judgment, have them make that judgment for themselves, as opposed to saying this algorithm is 0.8 AUC, go figure. And so this hundreds of training cases help them do that. Okay, so what happens? Good news. So first of all, look at this number. It's 60% accuracy. We've not done any oversampling. What does this mean? It means this is a really difficult task for people to do. And this is something that is recognized in the community of those who do this task. And we can boost that up by from 0.6 to 0.65, which is pretty good. And importantly, we have a much improved sensitivity going from 0.7 to 0.72. All right. But what was surprising was really our next result, which is that if you let the algorithm just work by itself without having any clinician intervention, it actually gets to an accuracy of 0.79. Specificity goes up to 0.87. And this is crazy. I couldn't believe these numbers. Why is this going on? Is this some sort of an anomaly in terms of our results? Now, since we've published this paper, there have been about 3 other pieces of research that have similar findings, that actually, if you had the algorithm working without clinician oversight, it would do better than with clinician oversight. So what's going on here? Let's break it down by looking at individual people. So here we have unassisted physicians in gray, and we have assisted physicians in blue, and we have the model performing by itself on these X's right here. So you can see, first of all, no physician actually surpassed the performance of the algorithm by itself. Okay, some physicians got worse. All right, some physicians got almost to the level of performance of the algorithm, probably by trusting every time what the algorithm was saying. Now, there's something to be understood here in terms of mistrust in the algorithm's output or overconfidence in one's own diagnosis. There was also another explanation here that when the algorithm was outputting probability buckets, if it was right in the center, you wouldn't be able to tell if it was less than 50% or greater than 50%. So it's also about the fine-grainedness of the algorithm's output. But I really want to point our attention to that. This is not a straightforward problem of being able to incorporate algorithm predictions into decision-making or into the predictions of physicians. This expectation is really misguided. And I think for us to be able to understand this, we have to take into account what are the experience levels, what is the interaction format, what is the case difficulty, and what are automation bias factors that we need to try to understand. These are the four pieces I think we need to really try to tackle over the next few years before we try to integrate these systems in scale. Cool. So that's the first direction. I want to talk about the second direction, which is real-world medical data curation.
Pranav,
yes.
A couple questions from the audience.
All right.
Before we continue, first, in most of the studies that you have been describing so far, there is a ground truth, or there is, some sort of real diagnosis associated with the cases. I guess it depends on each, type of study or each modality. For instance, in the latest case, you mentioned the microbiology test. And in the case of ACLTRs and others, how do you guys, determine the truth? Is it by a committee of, experts, or is it with a follow-up test or something like that?
Yes. So there are two broad categories, as you correctly alluded to. The first one is confirm the test with the results of another test. So for the pathology example for HCC-CC classification, which is two liver cancer subtypes, we're looking at immunohistochemistry to be able to determine the standard. Of course, this is not a resource available in a lot of settings where this decision has to be made. What often is the difficult part about using the results of a different test to be able to determine the label for the index test is that sometimes the other test is only available for certain cases. Example, if you have an X-ray suspicion of pneumonia, then you might get a CT scan to confirm it, but you're not going to have a CT scan associated with every single chest X-ray. So in that case, you only have access to the gold standard here when there is a determination made by a clinician that we must seek this additional test. So this often doesn't work in a lot of setups. So for chest X-rays, for instance, there are two things we know that we leverage. One is that chest subspecialty radiologists are a lot better than general radiologists. So let's have them set the ground truth. And also that a consensus is much better at agreeing with another consensus than as an individual radiologist. So let's try to have a consensus. And so broadly, I would say the two methods of setting ground truth here are a consensus of specialty medical experts, and the other one where it's the result of another test.
Great. And when you are training the models that are assisting physicians, are you using, labels that are the ground truth, as you're describing, like depending on the case, or what is the ground truth used for training, basically?
The ground truth used for training is often very different than the ground truth used for testing. For radiology, one source of that truth, which is used for labels, is some sort of a way to extract those labels from radiology reports. So that's often what we do. Manual annotation is often not a luxury that we can afford in resource-constrained environments. But when it's necessary, we do it. Fun fact I should mention. So for the head CT example I showed, where we had a model ultimately segment aneurysms. On the test set, that was done by a board-certified neuroradiologist, and I think it was actually a pair of them. On the training set, it was done by a group of 2 master's students in computer science who learned off the internet and through spending 2-hour-long sessions with the neuroradiologist of how to go about trying to do this task. And then I remember there was a weekend where they were just playing some music, had a monitor, and were just labeling these scans. So different sources, but often not the same as what's used for the test set.
Okay. And the last question is, accuracy is a gross measure, and false positives and false negatives may carry different clinical weight. So decisions making should not count them equally, right? Or how are you considering that in the results that you're presenting?
Yeah, I think the trade-off between what's required of a test in a setting is determined a lot by the context in which it's being used. The reason we went after accuracy in those particular examples is that the sets have been set up in a way such that we have about equal prevalences of positive and negative, which is when accuracy makes most sense. And I'm using accuracy here as a way to simplify our results, but if you care about the breakdown of your PPV, NPV, if you care about the sensitivity and the specificity of the test, they're all reported in those studies.
Okay, cool. That makes sense. Thanks, Pranav.
All right, cool. Thanks for those questions. So moving on, I think it should come across as no surprise to most people in the audience that medical data set curation is hard. It requires partnerships with hospitals. It requires IT frameworks for de-identifying and pulling data. It requires expensive manual annotation a lot of the time. And just as an example, when Google did their diabetic retinopathy work, they actually had residents label, it was between 50,000 and 300,000 examples. They had 50 residents, excuse me. And that's a lot of resources. But luckily, it's not been the kind of resource access one needs to do quality work in this space. And one of the reasons for that is there are creative ways of labeling. Here's an example. So this is a data set that I would argue really moved forward the chest X-ray interpretation with AI field ahead. And that was the NIH's release of chest X-ray 14. And what they did was they said the way we're going to annotate a bunch of chest X-ray images is look at their corresponding reports. We're going to build a report labeling NLP system and determine whether a disease is present, absent, or uncertain based on that evaluation of the report, and label it as 1 if it's present, or if it's absent or uncertain, then label it as 0. And then they made that data set public. It was 2 orders of magnitude bigger than anything that had been released previously. And the data set came out and it was criticized for its high label noise. This was a famous blog post at the time that is trying to circle disagreements to the label. And here you can see out of the 6, 12, 18 examples, there are more red squares and rectangles than you would like in terms of whether or not this particular set of X-rays has, I think, atelectasis, what was being evaluated. And it was clear at the time that there was a need for improving the system to be able to improve the labels and thus to be able to perform better in terms of learning. And so that's the task we set out to accomplish. And this task can be broken down into really 2 components. One is I need to understand what all the different diseases or conditions are and how they can be mentioned. And then I want to understand certain attributes that I want to associate with those mentions. The two attributes that I care about most is whether it's uncertain, whether its presence is deemed as uncertain. And an example here is purple. So this often happens when radiologists say it could be A or it could be B. And then second association that I care about a lot is negation, because it's a big difference to say there is consolidation versus there is no consolidation. And so these are the two relationships I want to capture first. And what I want to produce here is a structured output, as you can see on the right, where for a bunch of what we call observations, we associate them with 0, 1, or use. Cool. And so we were able to apply this labeler to a large data set of chest x-rays and associated reports from Stanford. And then we were able to apply it to a data set that was coming out of MIT, Harvard, and Beth Israel at the time, and showed that even on this data set, we were able to extract these particular diseases and these particular attributes with high accuracy. And so we released, once we had this labeler, one of the largest data sets and chest x-ray competitions to the world to be able to track progress on different chest x-ray interpretation tasks. And this is something that we also did for other tasks, including evaluation of fractures on bone x-rays, evaluation of abnormalities on knee MRIs. And this is a great way, as some of you know who have released large benchmarks for biomedical imaging, that this is a great way of being able to encourage advancements on these very key tasks. And it's not just about algorithmic advancements, it's also about being able to make effective investigations onto whether there is bias in these data sets, whether there are things we need to be worried about or taking care of when thinking about the risks of building AI models using data sets that are similar to this, that have been the focus of ongoing investigations and will be hopefully the focus of continued investigations as we're trying to get these systems into practice. Cool. Over the last few years, there have been a lot of teams that have competed on this leaderboard, and that's allowed us to understand what are key modeling insights that help improve performance. And there have been insights in terms of what loss functions are useful in terms of the incorporation of the hierarchy that is common in a lot of medical tasks, and the incorporation of uncertainty, which is very important to be able to handle labels where it's not just binary outputs, but there's also an uncertainty label that's existing in the data set. Okay, but here's what I want to argue. I want to argue that we've been building toy tasks all this time. And yes, there's been a lot of advancement, but we're still pretty far away from clinical deployment because we haven't designed data sets correctly. And here are 5 reasons that I think we have a gap. One is we have poor coverage of diseases. Right now, we have about 14 diseases that were labeled for in 5 of the largest chest X-ray data sets. And this is really not representative of what it's going to take to be able to do any disease from chest X-ray interpretation. Second is there's poor heterogeneity in patients. If you look at a lot of data sets that have been released, you're typically looking at a US population. You're typically looking at very specific clinical contexts. And unfortunately, without clear understanding of the exact clinical context under which the data was acquired. There's also poor generalization of the data sets that we're collecting across clinical workflows. This is not only true in terms of the technology used to acquire certain images. This is also true in terms of who makes what decisions based on what information in different parts of the world. If you look at a clinical workflow here in Boston, the number of people who take a look at an ICU film to be able to make a determination and the workflow is very different from what's going on in the majority of the rest of the world. A lot of tasks that we've set up really don't use all the clinical context that's available to the physician. If you ask a physician what they do when they look at a chest x-ray, they're going to say, well, I have a prior based on what the patient, based on what the patient is presenting with. And really the chest x-ray is there to confirm my prior or to rule out certain priors I have. But really, this is not the way we've been setting up chest x-ray interpretation tasks or a lot of other tasks. And no use of priors. Here, I want to not only talk about clinical context, but also what certain tasks in the real world that are done by doctors are, which is, here's what this patient looked like 6 months ago. I want to see if this patient got better or worse. But if you look at the range of tasks that are currently represented by data sets, most of them assume this is the first piece of information that you ever get about a patient, which is fair in terms of the ease of data set release, but really not representative of where we need to be. I want to give you an example of what we're doing to tackle a couple of these issues. So let's start with poor coverage of diseases. So earlier I mentioned the ability to extract about 14 or so conditions from radiology reports as being something that have been done by a large majority of data sets. We've been thinking, how can we extract all of the important information from, let's say, radiology reports or any text that we might care about associating with a particular patient profile? Here, we're looking at how we can expand the scope of tasks for radiology reporting by converting a document of text, here in this case, a radiology report, into a graph. And what this graph will do is represent what is the content of the report in a structured way, and at the same time, try to get rid of the style that is associated with individual radiologist reporting. So here, for example, there are 2 sentences as part of the summary of the radiology report. And on the right is the corresponding graph that we extract, which not only is able to extract that there is an infection which has been suggested by the finding of an opacity, but also we can associate that the opacity was located at the lobe, and lobe is modified by 2 terms, it's lower and right. So one, this allows us to be able to set up tasks in a much more interesting way, where I can now say, I want to take in an image and I want to output this graph. But also I can use this graph to be able to train algorithms differently, where I can now leverage all this information to say, okay, I now know that this is not just a possible infection. I know that this is in the right lower lobe, so maybe I can supervise my algorithm to pay attention to the right lower lobe of the lungs. And we've made this benchmark now public. So this contains about 6 million entities and 4 million relationships that were extracted from 200,000 radiology reports. And this is public as of a couple of months ago. I want to talk about another one, which is poor generalization across clinical workflows. So one shocking fact to me when I started out working on chest X-ray interpretation is that most of the world still uses manual chest X-ray films, analog, and they don't have digital workflows. There's someone who's actually taking a look at a chest X-ray film and is making a diagnosis based off of that. So we thought, how can we try to translate the technology that we're building for digital chest X-rays to analog chest X-rays? And we came up with an app that we put together just as a prototype to show how or whether this would be feasible. And the idea here is to increase access of this technology to non-digital workflows where arguably it's required more. Now, any sort of clinical workflow integration, I found, often requires new modules which are not useful for development. And here's an example. Photos of chest x-rays do have enough information to be able to make diagnoses. In fact, one surprising finding was that the way doctors determine what a patient has from their chest x-ray, if they don't have enough confidence in their own diagnosis, is they snap a picture and send it to a WhatsApp group of doctors. And we found this to be true in several developing countries. Oh, also true in the US. And we found that if we have this structure and we just put those images into the algorithm, there is a drop in performance. And yeah, we expected that. But these new modules can work with the introduction of new data sets, or at least that was our hypothesis. So we said, let's try to generate a data set of photos of chest x-rays by taking pictures of chest x-rays on different phones, and by also having synthetic transformations of chest x-rays to apply effects which one would find if you took a photo of a chest x-ray. And so obviously it's difficult to do photos of chest x-rays at scale. So we built a app where we were able to take tens of thousands of photos of chest x-rays from an Android phone, and then have made that data set now public. This test set also includes a real deployment setting in Vietnam where this is a workflow. In fact, what was surprising in these set of hospitals that I've been working with is that even though they have digital workflows, the way physicians interpret chest x-rays is get a printout of their digital chest x-ray and then interpret in analog fashion. As of just a couple of weeks ago, August 18th, that's more than a couple of weeks, just a couple of months ago, we have now made this an open competition, and you can already see that the top performing model on the leaderboard is able to outperform the best techniques that were working for digital chest x-rays. And I'm hopeful this will enable deployment into this new workflow format. Over the past couple of years, we've been partnering with an organization in Vietnam that has governmental support and has made this app that's an extension of what we worked on available for free in a lot of systems, and now have about 240 active doctor users across 63 hospitals or health facilities in Vietnam. Great. I want to talk about the third direction, which I think is necessary for us to be able to make this research question, make progress on this particular research question, and that's label-efficient algorithmic development. I just want to highlight that these are very real tools. At this point, there are over 100 FDA-cleared technologies, a lot of them doing tasks in radiology and cardiology. There are about 6 randomized control trials. A lot of these looking at being able to determine colon polyps in real time and being able to see if more colon polyps can be detected by using live AI. And there are now at least 2 CMS-covered AI algorithms where every use will generate revenue from the insurance companies. So this is very much happening. The trouble, other than that, we don't know how to evaluate the safety and reliability of all these algorithms that are being approved all that well, which is a different topic in itself. I think that one of the major difficulties is how we're going to go from the 50 tasks that we can do right now to the 5,000 tasks that we need to be doing to be able to enable scale progress. And right now, the reality of the matter is it's difficult and expensive to scale labeling for every task that we really want to be able to tackle. And I want to talk about 2 techniques that have been the bread and butter and which has been transfer learning for a long time, but now self-supervised learning techniques have made quite a bit of progress in recent years. And I want to cover some advances on these two fronts. So I'll start by talking about transfer learning. The idea for transfer learning is as simple as it is strange. Let's take a convolutional neural network that was first trained to identify whether an image has objects, has one of 1,000 objects that includes penguins, cats, dogs. And let's take that model and let's use that as an initialization for a model that's going to identify whether chest x-rays contain masses, pneumonias, or edemas. That's the basic idea. And what's been found to be the case is that pre-training on natural images helps. So here, if you see these two bars, you can see that pre-trained models generally outperform models that were initialized from scratch. And what we've recently shown is that it's actually pretty strange how this relationship works. So what's been true for a long time is let's take a model that was performing really well on ImageNet and let's apply it to our medical image interpretation task. And we'll assume everything that does well on ImageNet is going to do well for our task. And here, this is showing different dots, which is different models on ImageNet. What is their ImageNet accuracy and what is their CheckSpurt, which is a common chest X-ray data set accuracy. And what we expect is this line to essentially look like a diagonal line going up. We expect there to be high correlation between how we do on ImageNet and how we do on medical image interpretation tasks. And that's simply not true. So we're not hill climbing or not able to leverage advances in computer vision for image recognition, which a lot of the world works on, onto key medical image interpretation tasks. Of course, medical image interpretation is not just about 2D tasks. A lot of modalities are 3D. And one of the nice things has been that pre-trained 2D convolutional neural networks have applied well to 3D tasks. Here in this study, we had just 1,000 training examples of NeMRIs. And what we did was we looked at individual slices of the NeMRIs, passed it through a 2D ConvNet, which was trained on ImageNet, and then max pooled the representations to get a representation for the whole scan, and then use that to make a determination of whether it had some sort of an abnormality. And we found that using just 1,000 training examples, we were able to rival the performance of radiologists. So that's nice. One of the ideas that I got super interested in when I was doing my PhD was this idea of using a model trained on penguins and cats has been useful for a slice of a NeMRI. Why haven't we considered using videos to be able to help for scans? It's just as strange of an idea. And here was our attempt at doing so, is let's take a model pre-trained, let's train it on YouTube videos to identify actions. There's a large video action recognition data set called kinetics. And let's use that to initialize a model that will detect appendicitis from CT scans. And that was our idea. And so we had an architecture, and it was a 3D convolutional neural network based architecture. And we showed that with pre-training, we can get this huge boost in AUC, go from 0.72 to 0.81. And this was using only 400 scans. And oh, by the way, it doesn't matter which architecture we use. You can do this across different architectures, and you'll find that there is this significant boost by pre-training. So that was awesome. And we found that we could apply that to other tasks. Here we applied it to head CTAs. Here we applied it to chest CTs for evaluation of pulmonary embolism. And we found big performance boosts by using video pre-training. transfer learning is not only useful on the image side, it's also useful to be able to do NLP tasks like radiology report labeling. And if you look at NLP tasks, there has been a large body of work to be able to build rule-based models for different tasks. And these are what I call automatic labelers. And automatic labelers can help you give sort of a heuristic for what the label should be. And so the idea we explored here was, let's use that heuristic to first pre-train a model. And here we used a BERT model. And then let's fine-tune that on a small sample of manual annotations. And so the idea here is, I just need a small sample of radiologists to label a few radiology reports for the presence and absence of different conditions. And I see this huge boost in performance across different diseases that I want to extract from radiology reports.
Pranav, we have a couple minutes left.
Oh, okay, perfect. I think I should be able to wrap up in a couple of minutes. And here we can boost performance over existing rule-based systems by fine-tuning a small number of manual annotations. Great. So that's transfer learning. I think an emerging frontier for being able to perform well with small amounts of data is self-supervised learning. And self-supervised learning, the idea is, how can we use unlabeled data to set up a task which allows us to learn useful representations for the task we really care about. And we're going to have a pretext task, and we're going to transfer that knowledge onto our supervised task. What's an example of a pretext task? One task that's taking off a lot is contrastive learning. In contrastive learning, you say, here's an image, here is an augmentation of the image. So I might rotate it, I might flip it. And I'm going to say, hey, you know, these two have the same content. Let's try to have representations for these two images such that they have the same context, the same representation. And let's take two different images and let's make sure they're very different high dimensional representations. That's the basic idea. And because in the interest of time, I'm just going to jump over to the takeaways here. So this is the broad overview of what I, with several others, really hope to achieve in the next 3 to 5 years, which is start making these AI tools a part of routine clinical care through focusing on these different aspects. I'll just highlight that I'm very interested in increasing the number of people who are joining us in our efforts. And towards this goal, I've created a Coursera specialization on AI for medicine. There's a weekly newsletter that I co-edit for the latest AI for healthcare research, and also co-host a podcast where we chat with investors and entrepreneurs in this space to really understand what's going on the industry side. I'll just mention that all this work was done in collaboration, and I want to thank several students I had the chance to work with, 3 of whom you'll get to hear from very soon, and several faculty across different institutions with whom the work that I presented today was done. And that's where I will conclude. Thank you so much for having me.
Thank you for the invite. It's our great pleasure to be here this morning, and we're excited to share our work with you on contrastive learning of 12-lead electrocardiograms using physiologically inspired augmentations. So ECGs are a common non-invasive diagnostic test that's used commonly in clinical routine. And just, I guess, as a benchmark, according to 2016 data from the CDC, there were over 41 million ECGs that are performed annually in the US. And essentially what they are is a visual representation of cardiac function and activity. And so as the heart beats, we see that the electrical signal travels from the sinoatrial node to the atrioventricular node, and then the bundle of His, and finally the Purkinje fibers. And these voltage changes are then recorded and detected on electrodes that are placed at various points in the body. And you can use these for a variety of downstream tasks, ranging from as simple as a heart monitor to tasks that are more complex, such as arrhythmia detection. as well as identifying regions of damage from reduced blood flow or heart attacks, as well as structural abnormalities in the heart. And there are different kinds of ECGs, so the number of leads can depend on the task or setting that you're going to be using for. The work that we're going to present today is on 12-lead ECGs in particular. And really the aspect we want to focus on is on the nature of how ECGs represent the spatial and temporal activity of the heart. So as you can see on the slide, here we have the standard 12 leads of the ECG, as well as their placements on the body. And what we can see is that what the leads really represent are these projections onto 12 different spatial axes of the heart's activity. And they're oriented in such a way that at pretty much any moment, we have a complete picture of the 3D direction and magnitude of conduction. And this makes intuitive sense to us just looking at the diagram as humans when we're doing ECG interpretation because we understand how it's collected. But computer vision algorithms don't have this a priori understanding of the mechanism through which data are generated. So a natural question is, what are ways we can incorporate this prior understanding of the structure of the data to help models learn meaningful representations? So even without this understanding of the spatial temporal nature of ECGs, supervised learning has been shown to be able to extract clinically relevant features for ECG interpretation across various settings and tasks. So this is one of the first papers using end-to-end deep learning across a variety of diagnostic classes. And the task they did it for was for arrhythmia detection. And the author showed that the model that they trained was able to achieve an average F1 score across an independent test set that outperformed an average cardiologist. And this was in the ambulatory setting, so it was with a single lead ECG. On the right here, we have a similar study that uses deep learning to detect atrial fibrillation. But in this case, it was done using standard 12-second, 12-lead, 10-second, 12-lead ECGs. And finally, we have a study that uses deep learning essentially as a feature extractor to build patient-level ECG profiles. And they showed that using these representations, training traditional machine learning methods like gradient boosted trees on top of them, they were able to quantify cardiac structure and function accurately. And this was also done with 12-lead ECGs. And what all of these studies have in common, even though they vary across different settings and tasks, is that they were supervised learning and they required tens or even hundreds of thousands of labeled samples to achieve performance that would be clinically feasible to use. And as Pranav alluded to earlier in his task, when you have a bunch of different downstream tasks that are available, this can be a severe bottleneck when it comes to labeling, as it's especially labor intensive to label tens of thousands of labels for every single task and setting. And labeling is especially costly in medicine, but it's also a problem in other domains, such as computer vision. So thankfully, we have this large body of literature that we can draw on from other domains. And so self-supervised learning is one strategy to get around this bottleneck of labeling. And it's essentially what it is a form of unsupervised learning that exploits properties of a given modality to define pretext tasks. And now, like the first and most obvious benefit of self-supervised learning is obviously that you're able to exploit the volume of unlabeled data that's produced, which is generally much larger than the amount of labels that you're able to produce. And you require less manual annotations to learn meaningful representations. I guess for an example, in computer vision, working on the ImageNet data set, current state-of-the-art methods are able to achieve performance that are comparable to supervised learning with less than 1% of the data. But this also has a few non-obvious downstream consequences. For one, representations are not tied to a specific downstream tasks. So this naturally leads to easy feature reuse for transfer learning, as the features aren't targeted for a specific task. Also, representations aren't dependent on your class distribution because you decouple the representation learning from any specific classification task. So it's not as sensitive to class imbalance. And this also leads to a case for rare event prediction, which is especially relevant in medicine. There's this idea of few shot versus many shot learning that lends itself naturally to self-supervised learning. Within the self-supervised paradigm, Contrastive learning is one family of approaches that has recently risen to prominence in computer vision. And the intuition behind this is basically that some pairs of data points are more similar to each other than other pairs are, and that these similar pairs should have similar representations to each other, while disparate pairs should have different representations. So in this example, we would naturally expect that two different cats should have more similar representations, so we're going to deem them as positive views to each other, while a cat and a dog should have different representations, and we're going to call them as negative views to each other. And obviously, the whole point behind self-supervised learning is that we don't actually know whether the image is a cat or a dog. So instead, what we're going to want to do is generate pseudo-labels to define views without using actual labels. So various positive view definitions have been proposed for computer vision, such as different modal views of an image. But one of the most popular methods that we want to focus on today is called SimCLR. And essentially what they do is they define positive views as stochastic augmentations of the same image, while negative views are augmentations of different images. And so the key idea here is that we want to be able to leverage these invariances in images to produce semantically equivalent views of the original instance that we would expect to have similar representations. And we're going to enforce this through the loss function. So examples of augmentations that you'd expect to preserve semantic content are random rotation, cropping, color distortion, or Gaussian blur. And these are very modality specific, so it depends on the semantic context of the modality. So now we're going to turn our attention to some different ways that contrastive learning has been applied to ECGs. And this isn't meant to be a comprehensive sampling, but just a general overview of different approaches that have been made before. So the first one we're going to look at is more or less a straightforward extension of SimCLR to the ECG domain. So the authors propose to use SimCLR with 6 different augmentations. So we see here we have Gaussian noise, Gaussian blur, channel resizing, random resize crop, timeout, as well as dynamic time warping. And each of these augmentations more or less are directly inspired from either computer vision or standard time series analysis. And some of them don't make all that much physiological sense in that you would never expect to see this in a naturally produced ECG. But all the same, the authors show that pre-training with these augmentations in SimCLR significantly outperforms random initialization. However, as stated earlier, these augmentations don't really take advantage of the unique properties or nature of the ECG, but is kind of a direct extension from other domains. So the authors also propose another suite of transformations that can be used with SimCLR. And these are essentially physiological transformations. So they're meant to model realistic perturbations that can occur due to inaccuracies in the measurement process. So they propose using baseline wander, which is a low frequency artifact that can arise from respiration or movement of the patient. Next, they propose power line noise. So this happens when an ECG picks up interference from nearby power lines. Similarly, they propose electromyographic noise. So this is electrical noise due to muscle contractions. And finally, they use baseline shift. So this is from changes in electrode skin impedance because of electrode motion on the body. And so these augmentations are designed specifically for ECGs, and they're also able to show a significant improvement over random initialization. But they do fall short, however, in that they don't quite, I guess, leverage the spatial temporal nature of the data, as we mentioned earlier. Another approach that's been taken for contrastive learning is with patient contrastive learning. So the emphasis here is really on the clinical nature of how ECGs are generated. So the definition of positive and negative views focuses on patient identity. So as you can see on the slide, often multiple ECGs are taken from the same patient. So here, if patient A had 2 ECGs taken at different time points, we would expect these ECGs to have similar representations due to coming from the same patient, while ECGs from different patients should have dissimilar representations. And the authors are also able to show substantial improvements over random initialization, as well as over just using handcrafted ECG features such as heart rate, QRS duration, or T-axis. And they show this on a variety of downstream tasks, such as age prediction or sex prediction, as well as atrial fibrillation detection and left ventricular hypertrophy detection. The final, I guess, related work that we're going to be focusing on today is CLOCS, which stands for Contrastive Learning Cardiac Signals. And the authors propose a suite of methods that leverage the spatio-temporal nature of how the data are generated. So they propose three methods: contrastive multi-segment coding. So that takes advantage of temporal invariance, as on the left. And the idea is that within a short ECG clip, the diagnosis or rhythm is not going to change significantly as long as the clip is short enough. So therefore, different clips that come from the same recording should have similar representations. They also propose using contrastive multi-lead coding. So the idea here is that they see these different leads of the ECG as natural augmentations of each other because they're essentially the same cardiac activity projected onto 12 different spatial axes. And then finally, they propose using contrastive multi-segment, multi-lead coding, which is using both of these in combination. So you can imagine if we have a clip that has if we're looking at a 10-second ECG recording across 12 leads, for contrastive multi-segment, multi-lead coding, if we split that recording into two, we're going to have a total of 24 positive views because there's 2 segments for every lead and there's 12 leads, and all of those are going to be treated as positive views to each other. So kind of bringing it back to the spatio-temporal attributes of the ECG, as we can see, the unique attributes of the ECG have been leveraged in various ways, but we really wanted to focus on the underlying representation that the ECG is capturing. So if you think about what an ECG represents, right? It's essentially the 3D spatial activity of the heart project onto these different axes. And you can imagine that hearts have different orientation. Hearts can have different orientations or slight variations in structure. And these don't really matter in terms of clinical significance. They can have these very slight perturbations. And so the question that we were really trying to ask is whether we can take advantage of these invariances and that you can have minor spatial or temporal perturbations to model like natural physiological variation while using spatial temporal manipulations of the ECG to generate these different views for contrastive learning. And Bryan will be going over how we actually tackle that question.
Hey everyone, and thanks Ryan for the great introduction. I'm going to start off by jumping in and to discuss how we exactly leverage the spatiotemporal properties of an ECG in the augmentation setup that we use for our self-supervised learning setup. The main way we leverage the spatiotemporal properties of the ECG is by using a related cardiac signal, the electrocardiogram. Unlike a 12-lead ECG, which provides 12 2D views of the 3D cardiac signal, the VCG tries to show the 3D signal itself. All 3 VCG leads, X, Y, and Z, are orthogonal to each other, unlike the 12-lead ECG, where some leads are correlated with other leads. And the contours traced by the electrocardiogram reflect the actual propagation of the heart signal itself. And one good thing about the electrocardiogram is that they're really easy to produce. So a really simple way to convert from a 12-lead ECG, which we've reduced to 2 leads here just for the sake of simplicity on the diagram, is to use a predefined linear transformation. So in this case, we apply the linear transformation called the inverse Stowers matrix to convert your 12-lead ECG into a electrocardiogram. There's no additional info needed beyond this. But I guess I would ask the question, why would we need this? And I want to motivate this by arguing that because VCGs represent the 3D propagation of true cardiac activity, it reflects much of the natural variability in cardiac structure and orientation as well. If you consider any random population of human beings, there's going to be some natural variability in heart size and orientation. But for the most part, this variability doesn't really affect the arrhythmias and cardiac irregularities that you may see in this population. As such, we propose applying 3D transformations to patient VCGs in order to mimic these natural variabilities. So this can be like a rotation to basically reflect the different orientations in a patient heart, or scaling to reflect different sizes in a patient heart. And so hopefully this will train our self-supervised learning model to be invariant to spatial properties that are really unnecessary for diagnosing ECGs. As such, we propose the following two augmentations once you've derived your patient VCG. The first is rotation. Again, this is a linear transformation that is applied on all three axes by random values sampled on a user-specified range. So given a VCG and three possible rotations, we rotate in a random order on all three axes to produce a final output VCG. And this is meant, once again, to mimic variations in orientation that you see across different patient hearts. The next augmentation we apply is scaling. This is, again, another linear transformation where we scale on all three axes by a random value, which is sampled uniformly on a user-specified range. This is to mimic the variations in heart size that you may see across different patients. And hopefully, both of these augmentations will reflect natural variabilities that you see in patient hearts. Once we're done applying both of these augmentations, we convert back from a VCG to an ECG. This is, again, using the inverse linear transformation of what we use to get to the VCG in general. So this is the Dowers transformation. But note that because we're going from 12-dimensional space to 3-dimensional space and back, this process of VCG conversion and back to ECG is a little bit lossy. We decided to go through with this because our task in this project was to diagnose on ECGs, but a good research question that we haven't really explored is what happens if you diagnose only on VCGs? So once we're done converting back to the ECG, we decided to apply some temporal augmentations. And the main one we stuck with was time masking. This has seen a lot of success in self-supervised learning for speech recognition. And we chose this for two reasons. The first is we did not really want any temporal augmentations that would change the actual structure of the beat itself. So something like time warping or time dilation would not really reflect a lot of the invariances of different diagnosis classes. And second, we realized in consultation with a board-certified cardiologist that for most diagnoses, you only need one beat on one lead. So we decided to mask off significant portions of our ECG samples to train our self-supervised learning model to get good quality representations from low beats on dispersed across many leads. And so finally, putting it all together, our full augmentation pipeline is as follows. Given a 12-lead ECG, you convert into a VCG, apply a rotation, scale, convert back, and time mask. So with that in mind, I'd like to talk about our next contribution, which is a per lead view formulation. So to motivate this concept, I want to take a minute to talk about one of our major goals in designing this model. So even though 12-lead ECGs was diagnosing on 12-lead ECGs was our main goal, we wanted our model to be applicable even when all 12 leads are not available. So this could be in the ambulatory Holter setting, in an ambulance, or any other setting where you may not have all 12 leads. And so to do this, we constructed our model in the following way. So given an ECG that's gone through our full transformation pipeline P, we created our encoder architecture F beta to only accept one lead as input. So what this functionally means is for every 12-lead ECG that's passed in, you actually get out 12 per lead embeddings. And we did this, again, to reflect situations where you may not have all 12 leads ready. So the question naturally arises, like, what should a positive view be in the context of a one-lead encoder? So what representation should we be pushing together in the embedding space, and what representation should we be pushing apart? And an easy way that you can sort of sidestep this question is just by aggregating each augmentation's lead embeddings. So one simple way is to just, for each augmentation's 12 lead embeddings, you average and collate all of them into a final output embedding, and use your contrastive objective on the one pair of representations generated by each augmentation. So in our experiments, we denote this to be SimCGA, where the A stands for average. And this is fine. And in fact, many instance discrimination setups for computer vision actually do specify that you should only be using one pair of output embeddings for your contrastive loss. But it ignores a property of ECGs that we've seen before and that Ryan motivated in the introduction. It's that ECG leads are different views into the same cardiac activity. So these 12 ECG leads are one correlated and two represent the same underlying 3D signal. And so we argue, much like the authors of Clocks did, that leads can actually be thought of as augmentations of each other. And averaging per lead embeddings discards a lot of the semantic value of each per lead embedding. So to combat this, we propose a method that we're denoting SimCGL, where the L stands for leads. And unlike SimCGA, no correlation is actually done for each of the per lead embeddings. And instead, we calculate the objective function over each pair of lead embeddings over both augmentations. So intuitively, we're basically pushing all representations of all leads of all augmentations closer together. And this is much more physiologically inspired and has been seen in a previous work before. However, this work didn't really leverage a strong space of augmentations. So we're going to compare SimCGA to SimCGL later on in our experiments, but the final contribution that we sort of make in our proposal is patient-aware positive views. And I want to motivate this by stating a fact that all of us know, and it's that 12-lead ECG recordings can be long. So when we were processing the PhysioNet 2020 dataset, which is what we conducted our experiments on, we ended up taking an exhaustive set of disjoint crops. And we ran into this issue of the fact that once you make these crops, you kind of lose a lot of the per recording relationships that a lot of these crops have. And the question we wanted to address is, how should we consider the relationship between embeddings that come from different crops? So under traditional instance discrimination setups, augmentations of different crops coming from the same recording would be considered negative views of each other. This kind of makes sense because even though you're performing augmentations, the underlying data that you're performing augmentations on is not the same. And so traditional instance discrimination setups would view these as negative pairs. However, given our downstream diagnosis classification task, this doesn't really make sense because these crops are all because crops that come from the same patient are all tagged with the same downstream diagnosis info, they're guaranteed to classify to the same set of diagnoses in the downstream task. So instead of treating different crops from the same patient as negative views, we instead propose treating them as positive views. This is in line with similar work done in patient contrastive learning, and it basically treats each crop as positive views, so you never really push representations of crops from the same patient apart. And it makes much more sense given the downstream task that we're trying to minimize over. So finally, I want to formalize a lot of the stuff that I've talked about today by showing the full objective function. There's a lot of math here, but there's three main things I want to focus on. And all three of these codify our main contributions and changes over traditional SimClear. So the first deals with the set of all positive view pairs P. So what do I mean by this? We've seen two methods that reflect different ways of considering positive views. The first, SimCGA, considers positive view pairs as full 12-lead augmented ECGs. So in SimCG-A, your set P would just be each 12-lead ECG that you've performed an augmentation on. And the second, SimCG-L, considers positive view pairs as each per lead pairing over all 12-lead ECGs. So this is a much bigger set. And basically, this distinction informs a lot of the way you compute the loss function. Because you're computing an objective over many more pairs, positive pairs, we wanted to reflect that in the objective itself. So the second main part of our objective function is an indicator condition C that we put in to evaluate true to true if and only if data samples, in this case I and K, are positive views of each other. So what this translates to is what we just talked about in our patient-supervised contrastive loss, where we treat all ECGs from the same patient as positive views of each other. And the final part of our problem formulation that we wanted to discuss are the output embeddings Zi and Zj themselves. No matter how you're collating them, each embedding was produced by 12-lead ECGs that went through our full space of augmentations. So this is the conversion to the ECG, rotation, scaling, converting back into ECG, and time masking. So, and with that in mind, I just want to make one notational thing. We denote 3 kg, which is our main proposal, to be the following: SimCG-L, so that is our per lead formulation; patient-aware loss, which is considering different crops from the same patient to be positive views; and our full space of augmentation, so that is rotation, scaling, and time masking. And with that in mind, I'm going to hand it over to Gautham, who's going to talk about experiments.
Thank you, Brian. So before we dive into experiments, I'd like to first talk about what our motivation is with these experiments that we propose and go through. So our first motivation is to find good views for contrastive learning with our experiments. And I guess that leads to the research question: what is a good view for contrastive learning? And in the paper, What Makes for Good Views for Contrastive Learning, authors argued that a good view retains task-relevant information while reducing mutual information. So at the left, you'll see that we have an image and 2 augmentations. We feed them through the encoder and we try to capture as much information about the stimulus as possible. That's this general contrastive learning setup. And the hypothesis is that we want to allow the information between these 2 augmentations to be at the sweet spot, which is the perfect amount of information to just have enough information to do the classifications. So at the right, you'll see that on our x-axis, we have the information between these 2 views. And if that information is too much, then we'll have extra noise. This could be mutual information. And if the information is too little, that could be a less task-relevant information, then that might not be enough signal. And in the middle, you see that sweet spot, which is the perfect amount of information needed to make our classification. So that's one motivation. And we'll dive into this motivation for our ECG signals. With ECG signals, as Brian mentioned earlier, we have our VCG transformations, but our goal with these VCG transformations is to rotate, is to basically imitate the variance of the heart position and sizes in humans. With our rotation transformation, we are imitating the variance of the heart position and hoping to reduce that mutual information between ECG signals. And when we scale, we are hoping to reduce the mutual information that stems from different heart sizes in humans. Our second motivation in our experiments is to combine augmentations for contrastive learning. And the reason for this is that the paper, A Simple Framework for Contrastive Learning of Visual Representations, found that, especially for self-supervised learning, the combination of augmentations has greater performances than any single augmentation. So in this diagram, you'll see along the diagonal, we have single augmentation, we have models with just single augmentations. So at the top left, we have a cropping of just one augmentation. And across the diagonal, the performances are lower than the outer sides of the diagonals. And this is because those performances are with multiple augmentations, whereas that diagonal is just one augmentation. And the ultimate argument is that with self-supervised learning, especially for self-supervised learning, a combination of augmentations will lead to better performances. So that's our second motivation for experiments. And we will try to combine augmentations once we find the best augmentation strength. So our first augmentation that we'll be going over is our rotating, and this is with SimCGA. So if you recall from Bryan's definitions, that's SimCGA. And all these experiments were done with linear evaluation in our contrastive learning setup. So as you'll see, as we increase our rotation, the number of degrees for rotation, and this is basically when we rotate, for example, if we say we're rotating for 5 degrees, that means we're sampling uniformly from -5 to 5 degrees. And for 25, it's sampling uniformly from -25 to +25 degrees. But as this range increases for the distribution, we find that our performance is strictly improving. And we intended to see, or we wanted to see that performance, we wanted to find a sweet spot essentially. And we predict that we might not have been able to find a sweet spot because we haven't checked enough parameters for our rotation. So one thing that we'd like to do in the future is increase the amount of rotation and look at the results from that. So we do only have 4 data points here, and we want to increase the number of data points and the range. Next, for our single augmentation of scaling, we do find that our best performer is in between the lowest and highest scale factors that we tested against. So our best performer is at the scale factor of 1.5. But again, we only do this for 4 different scale factors, and we hope that we can do it for more in the future. But yeah, at this point, we do see a kind of U shape from 1.05. We have at the 1%, we have 0.771, and then it increases up to the scale factor of 1.5 and then decreases again. So there's a U shape. And if you think about the slide I showed earlier, the augmentation strength directly correlates to the amount of mutual information and task relevant information in our views. So we predict that this might be a sweet spot. Now, as I explained earlier, we do want to combine our augmentations. So I'm going to go through each of the combinations and compare them with their respective single augmentation. So for our first combination of rotate 45 and time mask, we see that when we compare this to our best performer in just rotate, which was at 45 degrees, we outperform at every data fraction. So at 1%, we outperform with 0.812 versus 0.801. And at 10% and 100%, similarly, we outperform. So this kind of goes in line with what we thought about earlier, which is that the combination of rotate and another augmentation, time mask, is greater than the best performer rotate. Similarly, for scale, we see that our combination with time mask is better than the best performer for scale with a single augmentation. So our best performer was at the 1.5 scale factor, and you'll see at 1%, we have 0.798, whereas the single augmentation was 0.795. And similarly, at 10% and 100%, we outperform scale. So this is, again, boding well with what we intended to do earlier, which is combining augmentations. Finally, for rotate 45 and scale 45, we can compare against the best performers for rotate and scale individually. And we do see that at the 1%, 10%, and 100% label fractions, our rotate 45 plus scale 1.5 beats the best performer for rotate at 45 and the best performer for scale at 1.5. Finally, our best combination, which is rotate 45 plus scale 1.5 plus time mask, similarly beats the single augmentation for rotate and the single augmentations for scale in the same manner that rotate 45 plus scale 1.5 beat the single augmentations. So across all 4 combinations, we see that it is strictly better than any single of the combinations used earlier. So now I'm going to compare our methods, SimCGA plus VCG plus time mask, SimCGL and 3kg, to the baselines that Ryan kind of brought up earlier. And in these comparisons, I'm just going to be touching on the differences between their method and our method, instead of, because I know Ryan has already talked about what these methods actually do. So for patient contrastive learning, or PCLR, the major differences between their method and our method is that they use a 12-lead encoder, whereas our encoder was a single lead, and they don't have augmentations. And because they use a 12-lead encoder, they can't have per lead views, which is one pivotal part of our process. And we see that at all the label fractions, all our methods outperform PCLR. Yeah, and once again, our methods are for linear evaluation. So that's something to keep in mind. Next, I'm going to compare to clocks. So I think Ryan already mentioned this, but I'm going to quickly go over what CMSC is, which is when you have positive pairs from the same lead, but different segments. And then CMLC is when you have positive pairs at the same time, but across different leads. And CMSMLC, which is the third self-supervised baseline, is a combination of both of these ideas. So once again, the major differences between clocks and ours was that they had no MLP. And we also found in their code base that there was no loss accumulation, no embedding normalization, and no augmentations, because their way of defining positive pairs was across leads and times. So in this example, all three of our methods, once again, outperform clocks at the 1%, 10%, and 100% labeled data fractions. Next, we're going to be looking at a comparison to another SimClear method. So this is the major differences were the 12-lead encoder, which is what this method uses, and the different augmentations. And also because they use a 12-lead encoder, they could not have per lead views. So yeah, the primary differences are strictly with the augmentations and encoder. And we find that the first method, SimClear plus RRC/timeout, that is a random resize crop augmentation and timing out some parts of the ECGs. We find that our methods outperform that at all three label fractions. Similarly, Ryan mentioned this, they have physiological noise augmentations, which are stemming from computer vision augmentations for signals. And for these perturbations, those were their augmentations. It was a combination of different augmentations. And our methods outperform the SimClear plus physiological noise at 1%, 10%, and 100% as well. Next, from the same paper, another method was proposed. I had a 12-lead encoder as well. And the only difference is that there were no augmentations, but they added an LSTM at the end of the encoder. And the goal for this LSTM was to predict a few time steps forward. And they believed that this would allow representations to be learned that were strong. And we find that with our methods, we outperform this CPC at 1%, 10%, and 100%. Finally, this method, signal transformation, was not a contrastive learning setup, and it used augmentations from computer vision. So I'm going to quickly touch on it. We beat them at 1%, 10%, and 100%. So now I'm going to talk about our, we finished looking at comparisons to past baselines, and now we're going to look at improvements that our self-supervised method has made over a fully supervised random model. So this graph here is on the y-axis, you'll see we have the AURC improvement. So this is the improvement of 3 kg, which is our proposal and our best method. This is the improvement of 3 kg over a fully supervised random model at the 1% label data fraction. So this is where we believe self-supervised learning has the greatest effect at the lower label fraction. So we look at 1%. And we find that, first of all, I'd like to point out that at every class, we are statistically significant in outperforming the fully supervised model. So we do outperform the fully supervised model at every class. But we see that in the places we make the best gains are typically of abnormality types of a rhythm abnormality or a conduction abnormality, whereas the areas that we make smaller gains are with single lead waveforms or multi-lead waveforms. Obviously, this is not strictly true. Like you can see for sinus arrhythmia, it is a rhythm abnormality. So we're not making any strong arguments here. We're just pointing out that these are the results that we've arrived at. And these abnormality types were classified by a certified cardiologist. But yeah, this is just something we came across. There are some exceptions, but mostly we find that rhythm and conduction abnormality types, we make the greatest gains with self-supervised learning, and we make smaller gains with multi-lead waveform and single-lead waveform abnormalities.
Hey everyone, I'm back to discuss some final research directions that we wanted to share for ways we could extend this project. The first one is the evaluation of self-supervised features on different downstream tasks and settings. As Ryan alluded to in the introduction, a lot of the representations learned by self-supervised setups aren't really specific to any downstream task. And so we were wondering whether or not we could apply a pre-trained self-supervised model to different kinds of tasks beyond just simple arrhythmia detection. The second research direction that we wanted to take was few shot versus many shot learning. So this is kind of motivated by the fact that in our downstream task of arrhythmia classification, a lot of the low frequency diagnoses did not have many labels to begin with. And on a 1% mask, this number of labels drops even more. And so it appears that based on the fact that we outperformed a fully supervised baseline on the 1% label fraction, that self-supervised learning could possibly be used in a few shot setup to improve these small frequency diagnoses. The third research direction that we wanted to go for wanted to propose is exploring other 3D augmentations in pre-training. So we discussed rotation and scale, but obviously there's a bunch of other 3D transformations that, even though they may not be rooted in like natural orientations of the heart, could still possibly be useful for self-supervised pre-training. And the last one, which I alluded to in the methods section, was just training models directly on VCGs. We didn't really explore this because we wanted to stay true to like the ECG domain in particular, and also allow for one lead ECGs to be used. But training models directly on VCGs allows you to sort of circumvent a lot of the losses you gain in the conversion process for VCGs, and could be useful in its own self-supervised pre-training task. So with that in mind, I just wanted to thank everyone for listening to our talk, and happy to take any questions with Ryan and Gautham.